So what's the chemical logic or chemical rationale for TCA? Well, I've kind of alluded to it, but we should probably get into it a little bit more clearly. What the TCA cycle is doing is using equivalents of acetyl-CoA to introduce new carbon into the cycle and then see those oxidized to CO2. And that oxidation is where we get the chemical release. So fundamentally what we're doing is we have our equivalent of acetate, or acetyl group, and we're taking this acetyl group, and through the whole process of the cycle, I'm really converting it to a pair of CO2. And this is where all of this energy is going to be found. And recaptured in the form of NADH, FADH2, and GTP. Now, what this involves isn't just oxidation of the acetyl group, but involves a cleavage of a carbon-carbon bond, because we have a carbon-carbon bond, and typically you see two avenues or approaches in biological systems for cleaving carbon-carbon bonds. The first is through cleaving alpha-beta carbonyls. So what that looks like is if you have a carbonyl, you have your alpha carbon, or carbon alpha to that carbonyl, and carbon beta to that carbonyl. And you often see cleavage between the alpha and beta carbons, and you see this in, for instance, fructose bisphosphate aldolase enzyme mechanism that we saw in the previous chapter. Now, another approach, which you see quite often, is cleavage of a carbon-carbon bond via alpha cleavage of an alpha hydroxy ketone. And what that looks like is we have our ketone, we have a carbon alpha to that ketone, and we have a hydroxy or hydroxyl group on that alpha carbon. So this is an alpha hydroxy ketone. And you also will see cleavage commonly at the position between the ketone and that alpha carbon for this functionality. Now, and we'll see examples of this in, let's say, transketolase reaction in the following chapter. Now, neither of these strategies is really suitable for acetate, because if you look at the structure of acetate, let me do a redraw. We don't really have the features required for this. We don't have enough carbons in the chain to do a alpha-beta cleavage, and we don't have the structures required for cleaving a alpha hydroxy ketone. So what we see in the TCA cycle is a fairly sophisticated workaround where the acetyl group binds to the oxaloacetate, and following binding to the oxaloacetate, we carry out beta cleavage. So this is the chemical logic we have in the TCA cycle for adding a pair of carbons and cleaving those carbons and seeing those carbons oxidized all the way to CO2 to abstract as much energy as possible from them. So how is pyruvate oxidatively decoupled to acetyl-CoA? If we take a look at the structures again, pyruvate looks like this. And acetyl-CoA retains, excuse me, can't forget the carbon, the acetyl group, but replaces it with coenzyme A. Now, and this is executed via, via pyruvate dehydrogenase. Now, pyruvate produced by glycolysis is a significant source of acetyl-CoA in the TCA cycle. In eukaryotic cells, glycolysis occurs in the cytoplasm, and the TCA cycle reactions and all subsequent steps of aerobic metabolism take place in the mitochondria. So pyruvate must first enter the mitochondria to enter the TCA cycle. So that's the first element of control in this, is that we have a transfer of pyruvate from the cytoplasm to the mitochondria. Following that, we can describe the oxidative decarboxylation of pyruvate to acetyl-CoA as following. We can say that pyruvate plus coenzyme A plus NAD plus yields acetyl-CoA plus CO2 plus NADH. And this reaction is catalyzed by pyruvate dehydrogenase. And now here's the thing. Pyruvate dehydrogenase isn't just a simple enzyme. It's a multi-enzyme complex. So let's write that down. Pyruvate dehydrogenase complex, or PDC. And the pyruvate dehydrogenase complex is formed from multiple copies of three enzymes, which include pyruvate dehydrogenase, or E1, the dihydrolipamide acetyltransferase, E2, and dihydrolipamide dehydrogenase, E3. So we've got our E1 component, the pyruvate dehydrogenase. The E2 component, our dihydrolipamide acetyltransferase. Lipamide acetyltransferase. And our E3, our dihydrolipamide dihydrogenase. And all are involved in the conversion of pyruvate to acetyl-CoA. So additionally, the eukaryotic pyruvate dehydrogenase complex contains a unique subunit, which is termed the E3BP, that has no known enzymatic function. But we're not going to linger on that variation specifically. So the active sites of all three enzymes are not far removed from one another. So the product of the first enzyme is passed directly to the second, which is then passed to the third, without any diffusion of the substrate or loss of products into the solution. So the eukaryotic version of the pyruvate dehydrogenase complex is one of the largest known multi-enzyme complexes. It's just an astonishingly large complex with a mass of about 9.5 megadaltons when fully assembled. And this really provides, this massive size gives you some insight into the biochemical complexity of this transformation. It's really a tour de force of mechanistic chemistry, which involves all three of these subunits and 5 different coenzymes. So what I'm going to do is walk through providing an overview of what the PDC is doing, and without going into the real nitty-gritty of pushing electrons. The book does an excellent job of providing some extra insight into the mechanistic details, but for this we're going to keep it surface level mechanism. So let's go ahead and make a little bit of space here. So first off, let's redraw our pyruvate. So we have our pyruvate here. And what's going to happen in the first step is pyruvate is going to be decarboxylated and is going to be bound by hydroxyethyl TPP, which stands for thiamine pyrophosphate. And what that looks like is the following. You can say loses CO2 as part of this process, and we'll call this step 1. We have the thiamine pyrophosphate, or TPP, and it comes in and grabs the pyruvate, forming the following. Hydroxyethyl TPP. Now, what's conserved here? Let me grab a different color. The carbon circled in red are the carbons circled in red. And you can see, as part of this decarboxylation, we also saw a reduction at the carbonyl carbon in this particular step. Now, in reaction 2, we have, and this is, being executed by the pyruvate dehydrogenase enzyme. Or the E1 group. Now, the E2 group, wow, I'm tripping all over my words here. Dihydrolipoyl transacetylase. Enzyme component is going to do the following. We have this really cool swing arm mechanism. And the swing arm is just a protein which has this long lipid arm of lipoic acid. And this is not going to be carbon correct. So forgive the number of carbons. But this lipoic acid has a sulfur-sulfur bridge in it, which is drawn at the bottom. And this allows for the sulfur-sulfur bridge to be formed or to be opened as part of binding something else. And what can happen is this swing arm comes in, grabs the carbons from the pyruvate off of the TPP, and this looks like the following. Maybe I give myself enough space here. Let's see here. Barely. I'm going to have to make a little bit of room here. And you can see that we've now added the acetyl group to this lipoic acid arm. And this is now primed for the addition of the coenzyme A to the acetyl group. So this happens on the third step. coenzyme A comes in and adds itself, or is added to, the carbons from the pyruvate forming acetyl-CoA. And what this does to the swing arm looks like this, and then this is closed, regenerating the sulfur-sulfur bond in the lipoic acid. And this has to take place through active reoxidation with the investment of an equivalent of NAD+. So this is the high-scale overview of what's happening chemically in the multi-enzyme complex, which comprises pyruvate dehydrogenase complex. Now, I should probably actually include some of these other components here. This is the dihydrolipoamide acyltransferase. And the dihydrogenase. The dihydrolipoamide dehydrogenase components. So this is what the three components are doing in concert to convert pyruvate into acetyl-CoA. Now, once again, the book has excellent electron pushing level mechanisms, which I urge you to take a look at because it's incredibly interesting. However, for the sake of brevity in this lecture, we're going to move on. So the first reaction in the TCA cycle is the reaction of citrate synthase enzyme, and that's where acetyl-CoA reacts with oxaloacetate in a Perkin condensation, which is a carbon-carbon condensation reaction between a ketone and an aldehyde or an ester. And what that looks like is this. Let's say we have our acetyl-CoA. I'm going to draw the methyl with stereochemistry, just so it's easier to see the abstraction of one of the hydrogens forming a hydride. So we have our acetyl-CoA, and we have the body of the enzyme, the citrate synthase enzyme, and it has a basic group, denoted B with a lone pair and charge. Oh, we don't have to put the charge on there. That's redundant. With a lone pair. And what this does is it reaches out and grabs one of the hydrogens off of this methyl group, forming a carbanion. And I'm going to draw that over here as an intermediate. And this is extremely basic. And this highly basic carbanion can then do an electrophilic attack on carbonyl of oxaloacetate. So that looks like this. So we always get a backside attack where these lone pairs will come in. Let me, this isn't drawn perfectly. Let me move the lone pairs down here. And I'm going to grab a different color for pushing electrons. These electrons come in, do a backside attack on the carbonyl. This is going to push the electrons up onto this oxygen where they have a choice of either coming back down and reversibly kicking off the electrophile, coming back down, kicking off a leaving group, or they need to reach out and grab something else. And in this particular case, that's what we see, because we also have another group which has a base which is in its conjugate acid form with a positive charge, which is formed from step 1. And in step 2, the same group grabs these electrons, and let me redraw it. I'm going to do a really long interaction here because I made an intermediate. And what this produces is the following. It's a reversible reaction where we now have citryl-CoA intermediate and in this group we have the acetyl-CoA, and in this group we have the oxaloacetate. And then we get a second step with the addition of water and the loss of coenzyme A. And this gives us our citrate product. With 3 carboxylates. Now what's really interesting about this is that we have 2 arms to this. We have our pro-S arm, and we have our pro-R. And we talked a little bit last semester, and a little bit in organic chemistry, about prochirality, because this compound isn't chiral, but it is prochiral, because through subsequent reactions it will become chiral, and we want to produce a product which has a specific chirality. So it's very interesting application of prochirality in this particular case with citrate. Let me shrink this down a little bit, make a little bit of room so we can see the entire picture. Grab a different color real fast. And let's talk about energetics here. So the overall delta G for this is about negative 31.4 kilojoules per mole under standard conditions. This is fundamentally irreversible for all intents and purposes. Very, very favorable reaction. And this is important because mitochondrial concentrations of oxaloacetate are maintained at very low levels. So, based on Le Chatelier's principle, that wouldn't favor the formation of products, because you would think it would drive towards reactants in reversible processes. But in this particular case, because delta G is so favorable that it will drive the reaction towards products very aggressively regardless. So a couple other fun facts about this particular reaction. Citrate synthase is a dimer. Citrate synthase in mammals specifically is a dimer of 49 kilodalton subunits, and on each subunit, oxaloacetate and acetyl-CoA bind to the active site, which lies in a cleft between the 2 domains and is surrounded mainly by alpha helical segments. So the binding of the oxaloacetate induces a conformational change that facilitates the binding of acetyl-CoA and closes the active site so that the reactive carbanion of acetyl-CoA is protected from protonation of water. Just fun fact. In terms of regulation, NADH is an allosteric inhibitor of citrate synthase. Sorry for the... There we go. Fix this. So citrate synthase is the first step of the pathway of TCA, which makes it a perfect target for regulation. Why would you not regulate the first step? And it has a very large negative delta G, making it favorable. So, as might be expected, it's highly regulated, and NADH, which is a product of the TCA cycle, is a perfect allosteric inhibitor of citrate synthase, as well as in succinyl-CoA, which is the product of the fifth step. And succinyl-CoA is fundamentally an acetyl-CoA analog, so this also makes sense. So moving on to the second step, the conversion of citrate to isocitrate. Citrate actually poses a problem. It's a poor candidate for further oxidation because it contains a tertiary alcohol. And tertiary alcohols are highly resistant to oxidation because they already have 3 stable bonds around them, and they don't want to see that oxygen further oxidized. So the only way to oxidize them is by breaking a carbon-carbon bond, which is obviously not something that we want to invest the energy in doing in a biochemical scale. So what happens is we're going to isomerize citrate to isocitrate via econatase in a 2-step process, which involves econatate as an intermediate. Let's take a look at what that looks like here. But inspection of the citrate structure shows a total of 4 chemically equivalent hydrogens, but only one of these, the pro-R hydrogen on the pro-R arm of citrate, is abstracted by econatase, which is extremely stereospecific despite chemically 4 of the protons or hydrogens being identical. So let's discuss this further. For citrate, we have this carbon, this backbone. Maybe I can shrink that back down a little bit. And we have our 2 hydrogens here. We have our hydroxyl in the middle. And then for ease, we're only going to do the pro-R and pro-S designations on one of the 2 carbons. So this is the pro-S arm up here, and this is the pro-R arm of citrate. So once again, the enzyme specifically targets the pro-R hydrogen on the pro-R arm of citrate, despite the fact that these 2 hydrogens and these 2 hydrogens are chemically identical. To get the stereospecific form of isocitrate, which we can feed into metabolic cycle, we don't want to have a mixture of 4 different isomers of isocitrate. We want a single isomer of isocitrate. Therefore, the enzyme has the ability to differentiate between chemically identical hydrogens to produce a product which is stereospecific. So, how it does this is the enzyme has a basic residue, and that basic residue comes in and grabs the proton. When it takes that proton, the electrons from this hydrogen, pro-R hydrogen to carbon bond, break, and they need someplace to go, so they hop up here, forming a double bond. This exceeds the number of allowed bonds on the second carbon. So what happens is we need to break something, and what breaks is the bond between the carbon and hydroxyl. Those electrons move to the hydroxyl, and this facilitates the formation of an equivalent of water. And what happens is, reversibly, we see the loss of water. We can also get the reversible addition of water. And as an intermediate, we see cis aconitate, which looks like this. I'm going to draw in the double bond. No, don't need a note. Well, now I've dropped a note here. I'm not sure exactly how to get rid of it. So cis aconitate, which is an intermediate. And then we see a second addition of water across that double bond. And what this does is it adds the water back, but it puts it specifically on the opposite side, so the hydroxyl's on another carbon. And we're going from having the hydroxyl on a tertiary carbon, or tertiary hydroxyl, to a secondary hydroxyl, which allows it to be oxidized. So our final product is the following. Isocitrate. Let me rewrite that because that is terrible. There we go. Once again, this is going to be added to the R arm of citrate. So, a couple details about this reaction. So aconitase actually contains iron-sulfur clusters, which consist of 3 iron atoms and 4 sulfur atoms in a near cubic arrangement. Cysteine residues from the enzyme coordinate the 3 iron atoms. In the inactive state of the enzyme, 1/4 of the cube is vacant. Binding of iron as F2+, activates aconitase. This is a good example of the way metals function in enzymes to support electrons or high energy intermediates, which wouldn't occur in the absence of these metals. Now, despite this iron-sulfur cluster, the equilibrium for aconitase reaction actually favors citrate. So in equilibrium, we're going to have a mixture of citrate, cis aconitate, and isocitrate, and this is about 90% citrate, about 4% cis aconitate, and only about 6% isocitrate, because delta G is actually about plus 6.7 kilojoules per mole. So it favors the reactants in this particular reaction, but it doesn't cost anything. So what happens here is we're going to have a dynamic equilibrium system between all of these, and in subsequent reactions, when isocitrate undergoes the following reaction with isocitrate dehydrogenase, which is catalyzed in the first oxidative decarboxylation, which is highly favorable, this whole sequence of reversible reactions will get pulled through towards products via Le Chatelier's principle. So speaking of which, let's talk about isocitrate dehydrogenase catalyzing the first oxidative decarboxylation. So now that we have isocitrate, and we've successfully moved the hydroxyl group, so we have a secondary hydroxyl. We see the action of isocitrate dehydrogenase catalyzing the production of alpha-ketoglutarate. And of course, what we also see here is the production of NADH from NAD+, and the loss of an equivalent of CO2. So this reaction has a net delta G of about negative 8.4 kilojoules per mole. And this is sufficiently exergonic to pull the aconitase reaction forward effectively via Le Chatelier's principle, which is what we were just discussing. And this is a two-step reaction which involves the oxidation of the C2 alcohol to isocitrate of the isocitrate, and it follows by a beta decarboxylation that expels the central carboxyl group as CO2, just leaving alpha-ketoglutarate. So the first step is the oxidation, and the second step is the decarboxylation. And I don't think we need to get into the details of the intermediate specifically. I think it's more important to talk about the importance of the specific step. This is the first decarboxylation, where the first of two of the carbons that we've added to the chain via the acetyl-CoA have now been kicked off in the form of CO2, and we've recaptured some energy in the form of NADH. But specifically, this is also the first link of the TCA cycle to electron transport, because isocitrate dehydrogenase provides this first connection between TCA and electron transport and oxidative phosphorylation through the production of NADH. It's one of those really important connections between two metabolic pathways. And therefore, it makes an important point of regulation so that these two pathways can crosstalk and be co-regulated. So in this particular example, NADH and ATP are allosteric inhibitors, and ADP is an allosteric activator. So let me just type those in here real fast. Allosteric activators. This is not how you spell activator. Activator. And ADP. Excuse me. I am getting out of sorts here. That didn't make any sense. Inhibitor. Inhibitor. And it makes sense, because if we have lots of energy in the form of surpluses of NADH, surpluses of ATP, we want to turn down the commitment of making new energy, and we want to start steering resources into storage. So if we have a lot of ATP, a lot of NADH, we want to start inhibiting the TCA cycle. Conversely, if we have low levels of ATP and we're seeing a surplus of ADP, we are running low on energy, and we don't want to be committing our resources to storage events. We want to be producing more NADH so we can make more ATP. And that's the logic behind this particular allosteric regulation. So now that we've completed our first decarboxylation, this leads us right into the catalysis of alpha-ketoglutarate by alpha-ketoglutarate dehydrogenase producing succinyl-CoA. And to remind everyone what this looks like, we have our alpha-ketoglutarate dehydrogenase. And we have a couple things going on here, because we are bringing coenzyme A in, and we are bringing NAD plus in, and we are getting an NADH out, and we are getting a CO2 out through our second decarboxylation. And the carbon being removed is the one attached to the activated carbon with the ketone. So what this looks like as a product is succinyl-CoA. Note the only difference is that the CO2 equivalent has been replaced with an equivalent of coenzyme A through binding the sulfur. Now, mechanistically, this is incredibly similar to the mechanism in pyruvate dehydrogenase complex. The alpha-ketoglutarate dehydrogenase is a multi-enzyme complex as well, and it consists of an alpha-ketoglutarate dehydrogenase, another dihydrolipoyl transsuccinylase, and a dihydrolipoyl dehydrogenase. And we're not going to get into it in gruesome detail like we did in the first one, but the dihydrolipoyl dehydrogenase of this reaction is identical to that in the pyruvate dehydrogenase reaction, and the mechanism is analogous as well. So note that we tried to grab Brad. We are producing NADH here, and this NADH, like the prior reaction, is going to be moving towards electron transport and oxidative phosphorylation and produce a substantial amount of ATP. Additionally, this is also a favorable reaction because of decarboxylation being a favorable process. So now that we've discussed the two subsequent decarboxylations, we've brought on 2 carbons in the first step, and we have managed to decarboxylate 2 carbons from citrate, forming succinyl-CoA. And now that we have succinyl-CoA, what's left? Well, everything from this point forward is really targeted at regeneration of oxaloacetate to complete the TCA cycle, and once we've regenerated oxaloacetate, we are primed and ready to start all over again. I think it's notable that from this point forward as well, you're not going to see a whole lot of regulation, either positive or negative, of the rest of the cycle, because most of the really important junctions have been regulated and met already, and we're just trying to get back to the beginning. Now, just because we're trying to get back to the beginning doesn't mean we're done pulling energy out of the cycle, though. Because succinyl-CoA is itself a high-energy intermediate and is utilized in the very next step of the TCA cycle to drive the phosphorylation of GDP to GTP, in mammals specifically, which can subsequently be used to make more ATP. So let's take a look at that. So make a little bit of room. So we have succinyl-CoA. Let's switch back to black. And we're going to convert succinyl-CoA to succinate. And what's going to be involved in that is we are going to kick off the equivalent of coenzyme A. And when we do this, we're going to redraw the structure as succinate, and you can really see the loss of that equivalent of acetyl-CoA that we added at the very beginning. So now this is executed via the enzyme succinyl-CoA synthetase, and what we mentioned already is that this is actually coupled to converting GDP to GTP, and that this subsequently can convert ADP through the phosphate transfer to ATP. So, like we said, succinyl-CoA is a high energy compound which we want to pull every bit of energy out of before we regenerate oxaloacetate. So, mission accomplished here. We got to get rid of this coenzyme A as well. And once we've regenerated succinate, let's take a look at this really fast, actually. You can see that we have these two carbons are these two carbons, and we've simply added the oxygen back to this carbon and then brought it up in line and redrawn it as succinate, following the loss of coenzyme A. So that it's clear where everything is being drawn from. But we're still not done, because we have an equivalent of FADH2 and an equivalent of NADH left before we get back to oxaloacetate. So, following the formation of succinate, we're going to see the conversion of succinate to fumarate, which is going to add the double bond. And this is going to be executed through succinate dehydrogenase. And we're going to remember draw this as a trans conformer. With the carboxylates as far apart as possible. And, like we mentioned already previously, we're going to get an equivalent of FADH2 produced. In this step. And FADH2 is a lower energy reducing equivalent which functions analogously to NADH as part of electron transport and oxidative phosphorylation. However, there's less energy to capture in this chemical conversion between the single bond and the double bond, but we still want to make the most of it. So we don't want to lose it. And this really justifies the development of a biological mechanism for harnessing different levels of energy in the form of reducing equivalents, which is what we're seeing here. And now that we've got our fumarate, we're going to add water across that double bond. And this is going to give us malate. And this is going to be done by fumarase enzyme. Now, like I said, all of these reactions move along minus significant regulation. But just because they're not highly regulated doesn't mean they're not of importance. We're still pulling energy out of each step. And, additionally, I forgot to mention something important. Succinate dehydrogenase enzyme, which is executing the oxidation of succinate to fumarate, which we're seeing here, is actually a membrane-bound enzyme, which is part of electron transport as well. So this is membrane-bound, and it's the only enzyme in TCA which is membrane-bound, and it's also integrated in electron transport. It's actually identical to the succinate coenzyme Q reductase of electron transport. And in contrast with all of the other enzymes of the TCA cycle, which are soluble proteins found in the mitochondrial matrix, succinate dehydrogenase is an integral membrane protein tightly associated with the inner mitochondrial membrane. So it's an astonishingly important link between TCA and electron transport because they actually share a common enzyme and tie the soluble protein processes to membrane-bound processes. Can't believe I almost forgot that. And then moving along, now that we've got malate, we have everything we need to make oxaloacetate through the action of malate dehydrogenase. To produce oxaloacetate. And this is just going to be the oxidation of the hydroxyl to a new ketone. And with that, we get, because it's an oxidation, the production of a reducing equivalent, NADH, which will also feed into electron transport. And this gets us right back to the beginning again. Now, we're going to call this a wrap for detail in the mechanics of the TCA cycle, but there's a few more things I want to talk about. I think we're going to pick up the next lecture with, let's see, we need to discuss the energetic consequences of TCA as an overview, which is essential. We need to talk a little bit about how the TCA cycle or citric acid cycle provides intermediate for all of the different biosynthetic routes, because this is the center of primary metabolism. And it's not just about energy generation, it's also about how all of these components integrate into other aspects of primary and secondary metabolism to centrally act as a supply chain. We need to discuss anaplerotic or filling up reactions. How does this cycle compensate when things start to run low? And I think finally a review of how TCA cycle is regulated as a system. So we have another lecture coming up, but I'm going to load this one up first, and then I'll be talking to you guys a little bit later soon.